Hello, everyone, and welcome back to Bio 181, Chapter 21, Viruses. Viruses are what we call infectious agents, composed of nucleic acids, either DNA or RNA, that are enclosed in a protein sheath we call the capsid. So they aren't a cell. They don't have all of the components that we're used to a cell having. And for that reason, they are called acellular, which means they are without a cell because they lack a cell wall, they lack a cell membrane, they lack cell ribosomes, etc., etc. They are parasitic, meaning they propagate in other cells, and as such, they are not considered alive. They are more like a vesicle of genetic code that makes its way into cells and rewires cells to make more viruses or more vessels of genetic code. And as you can imagine, in order to do this, they must be quite small. Again, so they're very tiny, they're acellular, parasitic, and not alive. So, why aren't viruses alive? Most decisive reason is that they cannot complete their own reproduction. They infect a host cell with their genetic material. Essentially, that material gets sent to the host cell's machinery, such as the ribosomes, to make viral proteins, polymerases, to make more viral nucleic acids. As we learned in the very beginning of this course, the most basic unit for life is the cell, because it takes a cell to do all 8 things on this list. For example, we wouldn't call the genes encoding DNA polymerase as alive on its own merits, because it can't do all of the things on this list. It takes everything in a cell holistically to meet these 8 properties, basically. So, nor would we say the same about a virus that can't conduct its own reproduction or energy processing for that matter. This chapter breaks down the steps of viral replication into 3 steps. First, attachment and penetration. This is when the virus first makes contact and attaches the host to the cell membrane, and thereafter, when the virus or its genetic material enters the cell. Second step is biosynthesis and assembly. Therein, the host cell's replication machinery, such as polymerases and ribosomes, are directed to produce and assemble new viral components like viral nucleic acids and viral proteins. Think of it as like a hostile takeover of the factories of the cell. The third step is release, where the new viruses are released from the cell, sometimes violently through the bursting of the host cell. 3 important types of viruses we will be discussing are bacteriophages, RNA viruses, and retroviruses. Uh, phage means virus, and so a bacteriophage is a virus that infects bacteria. They have some of the most interesting phenotypes of all of the viruses. They appear as a syringe with legs and function in much the same way. The head is what houses the DNA. The head is made up of a protein called capsid and protects the DNA. The collar and whiskers help to attach the tail, which, like the barrel of a syringe, is through which the DNA is injected. The tail fibers and base plate are for attachment and recognition of a compatible bacterial cell and helps initiate the injection of the viral DNA through the tail and into the cell. Here we have the basic viral cycle. This is the lytic cycle, where we first begin with attachment and penetration. We can see the little uh, um, basically 6-sided depiction of a bacteriophage there, plus the little green squiggly is the genetic material being injected into the host cell. Once inside, the phage DNA is going to basically dedicate the cell into making more phage DNA and more of the proteins and other components of the virus and assemble them into new viral particles. And then the end of the lytic cycle is the lysis of the cell. So lysis means it's being destroyed. Um, and the release of, um, you know, the newly formed viral particles. The lysogenic cycle is the second method that viruses can use. It begins with the same step, attachment and penetration. There's an extra step here. So, in this step, the viral genetic material actually integrates itself into the host's DNA and becomes one with the host DNA. And later, when there are some sort of environmental signals that tell the viral DNA or viral genome that it's a good time to begin making viruses, after propagation of the cell, maybe, then it's going to do what's called induction. And this induces the production of viral DNA within the cell. So this begins the biosynthesis and assembly pathway and then the release of the viruses later. When a lysogenic phage infects a host, expression of its genes can alter the host's phenotype, and this is called phage conversion. For the most part, Vibrio cholerae, for example, is a harmless organism. But the strain responsible for cholera of this organism is infected with a lysogenic phage, and from which the genes encoding cholera toxin, which makes it incredibly harmful, are then therefore present in this organism. And that would be an example of a phage conversion. Checkpoint one. Phage lambda is a bacteriophage that infects cells in a latent manner, meaning that it's integrating its DNA into the host genome until induction takes place at a later time. And that's the latency there. Which replicative life cycle is exhibited by phage lambda? Let's now discuss the RNA virus. This virus does not carry its genome as DNA, but rather it keeps it in the RNA form, which is very unique from what we've discussed so far with all cells. All cells keep their genome as DNA in their chromosomes, but these types of viruses store it as RNA, which is where viruses in this category derive their name. This image is that of an animal virus. This is how animal viruses commonly occur and is responsible for the flu. It's called influenza. This is an example of an RNA virus. It has a lipid envelope, and that envelope is studded with these spike proteins called hemagglutinin and neuraminidase. And notice how the neuraminidase has an ACE at the end. So we're going to find out later that this is an enzyme. Okay. Inside of the envelope is the protein capsid that protects and houses the RNA, and the RNA and its associated proteins are called a nucleoprotein. Again, we have three steps with these animal viruses. First, attachment. So, influenza virus becomes attached to a target epithelial cell. An epithelial cell is like a skin cell, it might be, or a surface cell, it might be on the throat, might be in the sinuses, it might even be potentially in the esophagus or trachea or something like that. And then you have penetration, and this is where the cell is going to engulf the virus by endocytosis. Endo meaning in, cytosis, cyto meaning cell, so endocytosis. And then the virus uncoats itself, and the viral contents are released into the cell, and therefore the viral RNA can start relegating work to the cell. In step four is biosynthesis, where viral RNA enters the nucleus, where it is replicated by the viral RNA polymerase. So, this even tells the cell to produce the proteins for viral RNA polymerase to conduct its replication. And step five is assembly. So the new phage particles are going to be reassembled into full viral particles. In step six, we have the release, where new viral particles are made and released into the extracellular fluid of the animal. And the cell, which is not killed in the process, continues to make new viruses. And we can treat viral infections by targeting replication. So, antivirals target one of the stages of the viral cycle. For example, Tamiflu, it's also called oseltamivir, uh, blocks the ability of the new viral particles from being released. How does it do this? Tamiflu is a neuraminidase inhibitor. And the virus uses this protein to dissolve the membrane of its host cell so that it can exit the cell. And that's the enzymatic activity of neuraminidase. And thus Tamiflu prevents new viral particles from being released because it inhibits the enzymatic activity of this surface protein. Checkpoint 2. Amantadine is an antiviral drug that is used to treat influenza virus by binding to and inhibiting the viral protein M2, which is necessary for the viral capsid release to release its contents into the cell. What is the name of the stage of the influenza virus life cycle that is inhibited by amantadine? Now let's discuss host range. The hemagglutinin and neuraminidase proteins, much like the base plate and tail fibers, help to find the right host cell and dissolve the membrane to allow the virus entry. As you can imagine, this is an animal virus, and it doesn't want to try to infect the wrong kind of cell, or else it will not be successful. For example, many viruses may affect your pets but cannot get you sick. This range of cells a virus can infect is called the host range. H spikes are associated with a protein called hemagglutinin, and N spikes are an enzyme called neuraminidase. H spikes help the virus identify receptors on the host cell and therefore identify the right compatible cell. And N spikes break down the membrane and penetrate the host cell. There are many different varieties of H and N spikes, hence the many different varieties of HN viruses out there, like H1N1, H5N1, etc. And here we can see that H1N1 influenza can infect swine, if we see in the bottom right corner, as well as humans. H1N1 is also compatible for infecting human cells. And therefore, this particular influenza strain can go back and forth between swine and humans. Now, we can develop vaccines to viruses. Usually, we use something like the outside surface components of the influenza for our immune cells to recognize and then develop our own immunity to, because we can now recognize these outside viral particles as foreign and attack them when we have a later exposure to the flu. Over time, viruses mutate and evolve at a rapid rate, and this creates a never ending arms race between our immune system and the virus. Through exposure to a new strain, our immune system becomes trained to identify a new virus based on its outer proteins. So you imagine if your immune cells are going around your body looking for viruses, they're only going to look at the outside of that virus in order to identify it. It's like how we would look at each other and recognize each other by maybe our facial features. As a virus mutates, some of its mutations affect the outer proteins and change their appearance, which stymies our immune system. Therefore, a new vaccine must be developed each flu season based on the predictions of what is guessed to be the new dominant strain that year. Since scientists called epidemiologists, cannot always get this perfect. We can sometimes have bad flu seasons. The 2017 to 2018 flu season was the worst season on record. The vaccine strain H3N2 was predicted right, but the clade, so the minor variation, was predicted wrong. And over 90% of H3N2 flu was not protected against. So, seasons in which H3N2 is the dominant strain are historically bad because H3N2 often mutates in ways that make it a bad match for the prepared vaccine. The next reason is that flu viruses are studied and grown in labs by cultivation in chicken eggs, believe it or not. And H3N2 is more vulnerable to egg adapted mutational changes and the inevitable mutations that occur when viruses are grown in chicken eggs. And these changes mean that the viruses may poorly resemble the viruses in the wild, making the vaccines, which are going to be the little parts of these viral cells to train our immune system, not as good as training our immune system for the appearances of the wild strain that season. However, technology is always adapting to to new constraints, and Novartis is now growing their vaccine in canine kidney cells instead of chicken eggs to reduce this complication. And the vaccine is just as effective and can be made faster and avoids egg adapted changes to the virus. Okay, now let's discuss a virus that we're all kind of familiar with by now, and that is the coronavirus. What is a coronavirus? Coronaviruses are a family of viruses that include viruses that cause the common cold, as well as many other more serious ones that infect the tissues of the respiratory tract. It's an enveloped RNA virus, and it is about zero point 0.5 microns in diameter. So, to give you an idea of that size there, remember we learned that bacteria are usually about, mitochondria, the length of them are about 2 microns. So, these are incredibly small in diameter. And we can view them with an electron microscope. Um, we can't view them under a normal light microscope, which we've kind of talked to earlier in the semester. Um, they can be observed with the surface proteins, um, giving an appearance of a corona, which means a crown. So if you look at these images here to the right, you'll see the surface proteins actually, those little spikes you can see around the outside of the virus, which is pretty neat. And they enter and exit the body through the respiratory system and are transmitted through respiratory droplets, airborne particles, direct contact, or indirect contact. For example, maybe they get deposited on a door, a handle, or a phone, and then you pick these up. These are called fomites. A fomite is any inanimate object where you can pick up a disease. Now, there are 7 known strains with human hosts. 4 are causes of the common cold. Others include the Middle East Respiratory Syndrome, or MERS-CoV, severe acute respiratory syndrome, which is referred to as SARS, and then COVID-19, which is a novel coronavirus. And that stands for Coronavirus Disease 2019. And COVID 19 gets its name from being the coronavirus disease discovered in 2019. Now, the virulence, or how effective a disease is at spreading, is called its R naught value, R sub zero here. It represents the average number of individuals to which an infected person transmits a communicable disease. The higher average number of individuals, the more virulent the disease is. COVID-19's R naught is 2.0 to 2.7 people. So on average, one person will get 2 to 2.7 other individuals sick. This is much more contagious than influenza, which is one of the reasons it was such a problem and became a global pandemic. Influenza's R naught is 1.5, and measles, which spreads incredibly rapidly and is incredibly contagious, its R naught is 15. And finally, let's discuss retroviruses. The retrovirus has RNA as its genome instead of DNA, and a capsid protein protecting it. Outside of that is the membrane envelope, and proteins embedded within it to help with attachment and fusion with host cells. What makes retroviruses so incredibly interesting is the route of replication it takes. Let's follow this process. In step one, we have attachment. The proteins on the outside of the virus identify markings on the host cell, attach itself, and step two, enter the host. After entry, the virus does something extraordinary. It converts RNA to DNA. Now, everything we have discussed so far is of DNA being converted to RNA by RNA polymerase. But retroviruses have an enzyme called reverse transcriptase that makes DNA from its viral RNA genome. This is why they're called retroviruses. They can go back to RNA from DNA. And um, and this DNA is called a proviral DNA. The DNA then enters the nucleus and integrates itself into the host's DNA via an enzyme called integrase. From this position, it now hijacks the host's RNA polymerase to make HIV RNA transcripts to be used in the fourth stage of biosynthesis, where the viral proteins in RNA are packaged into newly assembled viral particles that are released in step 6. And yes, as I in this example, we're talking about a retrovirus that we're all familiar with, the HIV virus, which causes acquired immunodeficiency syndrome or AIDS. It's probably the most infamous of all retroviruses, and interestingly, HIV infects T helper cells, which are cells that activate the immune system response. So it goes right after the immune system. And this prevents the body from mounting an immune response and leads to the onset of AIDS, which stands for acquired immunodeficiency syndrome. Scientists use their knowledge of the different steps of the viral cycle to target HIV, including reverse transcriptase inhibitors, protease inhibitors that disable an enzyme called protease that is used in new virus assembly, and integrase inhibitors that prevent the virus from integrating into the genome of the host cell. However, reverse transcriptase is more likely to make errors when converting RNA to DNA than DNA polymerase is when they make copies of DNA. So, what sort of effects would this happen, this higher error rate? Um, well, this high error rate would lead to a very high mutational rate in HIV. And this leads to mutants that are resistant to drugs, such as the reverse transcriptase inhibitor. So, in this example, the area of reverse transcriptase recognized by reverse transcriptase inhibitor has an arrow pointing to it, where a resistant mutant has a slightly different shape in the enzyme, which renders the drug ineffective. And finally, checkpoint 3. Describe one difference and one similarity between a normal RNA virus and a retrovirus. And that is the end of chapter 21, viruses. Thank you all so much for being here, and I wish you all the best.